Hello everyone, and thank you for attending today's webinar, Translation of a Diagnostic Virus Neutralization Method to a Quantitative Relative Potency Assay for an Antiviral Antibody. I'm Tristan Free, Digital Editor of Biotechniques at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speakers, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please let us know your thoughts on today's webinar by tweeting us at @BioAnalysisZone using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, presenting for us today from Biotherapeutics, we are delighted to have Jeffrey Patrick, Director of the GMP Potency Team, and Jessica Weaver, Bioanalytical Project Manager. Jeff serves in the role of Director of the GMP Potency Team at Biotherapeutics. He has more than 25 years of experience in biopharmaceutical development, manufacturing, and testing, including upstream and downstream development and analytical support, GMP QC lab consulting, and biologics discovery and early phase development. He has broad expertise in method development, validation, transfer, and implementation in CMC manufacturing settings. Jessica has more than 14 years of experience working in both academia, CMO, and CROs in the North Carolina area, and brings with her extensive GMP regulatory knowledge. In her role at Biotherapeutics, Jessica works to manage a team of project managers, scientists, and analysts specializing in GMP cell-based potency assays. She is also experienced in method feasibility, development, optimization, transfer, qualification, validation, and GMP stability, and a lot of release testing. We invite the audience to submit any questions you may have for our presenters throughout the webinar using the Q&A widget to the right-hand side of your screen. We will pose your questions to Jeff and Jessica in a live Q&A session after the presentation, and then follow up any outstanding questions offline. Thank you again for joining us today, everybody. I will now hand over to Jeff and Jessica to start today's presentation.
Great. Thank you, Tristan. Good morning, good afternoon to everybody, and thank you very much for joining us. We're excited at this opportunity to share some recent work with you, specifically on the development of a quantitative cell-based assay for the determination of relative potency of an anti-influenza antibody. This work's been done at Biotherapeutics in conjunction with our sponsor, Vistera, who's provided material and scientific input to facilitate this project. First, let's start off with a brief outline of what we're going to talk about. We're going to begin with some basic introduction of some concepts on neutralization assays, talk about viral therapies and neutralizing therapies and the assay methods and needs to support those. Specifically, we'll dial down a little deeper into the WHO test, which is a diagnostic test for influenza, H1N1. We'll discuss some specific objectives of the method in order to make it a GMP method and capable of product support. Then we'll get into the meat of the data and the meat of the project, the actual method development and transformation from a diagnostic test to a relative potency assay, including and finalizing with method qualification and some method capabilities, and then we'll wrap up with some conclusions. What you should hear today is about the transformation of a microneutralization method for detection of viral immune response into a method for relative potency for a neutralizing agent. Here it's specific to monoclonal, but in concept its general, being able to take a diagnostic neutralization method and translate that into a relative potency, and we'll talk about those specifics. We'll also discuss what steps were taken to optimize it, what our approach was, and how we did that to meet the needs of a GMP release assay for potency. And then finally, we'll talk about the validation or qualification of the method to current modern standards, including exploring accuracy, dose linearity, range, specificity, robustness, and precision, among others. So first let's talk about biologics and viral neutralization. There's a range of modalities that are explored and available currently, and they include vaccines, neutralizing engineered proteins, anti-serum, polyclonal antibodies, and monoclonal antibodies. They have some commonalities and some very distinct challenges in their development, but one thing that is in common is most of them require a potency assay, and that often begins with animals, as you can see to the right. There's challenges currently to try to expedite that and to reduce costs. Animal testing is both time-consuming and expensive. To address this, many assays are being developed towards cell-based assays, which reduce the costs and also will expedite the process. Specifically, this is to address the concerns around the 3 R's: reduce, refine, and replace, which has been pursued and supported by global regulatory agencies and others in the scientific community. In addition to the specific protein-related modalities that are specified here, vaccines, neutralizing proteins, and anti-serum, etc., this approach can also be used to approach gene therapies that ultimately lead to the expression of these proteins or similar in their modes of action. As I mentioned earlier, the specific modality that we'll look at here is a monoclonal antibody, again as a general and translatable neutralizing agent to demonstrate proof of concept. So what is a viral neutralization test? So here's a basic cartoon that captures the key points. In principle, it requires infectable cells, a neutralizing agent, and in this case for a diagnostic test, it's actually antibodies present in the serum, and then a source of the virus. The antibody in the serum or the neutralizing agent in the host cells can vary, as can the virus. Combination of the virus with the neutralizing agent, followed by incubation with the cells, results in a viral titer, which can be measured, and this is a determination of exposure or immunogenicity. The diagnostic assay that we're going to talk about specifically is the WHO assay for H1N1, which is shown here in the cartoon, where the heat inactivated sera are incubated with the H1N1 virus, then incubated further with MDCK cells, and we'll speak to those a little bit later. Following that incubation, an ELISA is performed against a nucleoprotein, and this is the determination of the readout in the viral titer. To adapt this general concept to a viral neutralization assay for relative potency, there's a small change. You still require infectable cells and the neutralizing agent. Here it becomes a monoclonal antibody or any of the other modalities we spoke about before, a source of the virus, and then one must add a reference standard. The same process occurs. The virus and the monoclonal are incubated, exposed to the host cells, and then a viral titer is determined. But now the reference standard is also a part of this determination, and that's what allows for the relative potency determination. Here it's depicted in a similar cartoon, with the principal changes being that the monoclonal is added instead of antibody in serum and heat inactivated serum. And the final readout is a series of titration curves which will allow relative comparison between reference standard and the samples of interest to determine a relative potency. This is where the power and the capabilities of this come. and again is generally applicable across broad opportunities. Let's talk briefly about the system that we're going to specifically use as a proof of concept here, and that's the virus first. H1N1 is a subtype of influenza A, also known as the swine flu. It's been responsible for several seasonal outbreaks and a historical pandemic, very similar to the one we're seeing right now with the coronavirus. This pandemic caused significant issues in 2009 with the outbreak, specifically around college campuses, and some business impact. And it's required a lot of investigation and investment to better control these outbreaks and provide sources of immunity, as well as therapies for infection. In the cartoon to the right depicts the virus itself. Of specific interest is the nucleoprotein, which is what will be used in the ELISA to actually determine titer and quantify the virus. The protein, or the neutralizing agent under study, is VIS-410. It's a monoclonal antibody directed to the influenza virus, H1N1. It acts by binding to hemagglutinin on the surface of influenza and actually neutralizes the virus. It has been shown to be successful in neutralizing viral activity and infection in challenge studies, but as we said before, animal studies can be quite complex, time-consuming, and expensive. So the move to create a relative potency assay was a strong drive for this project. And again, here it serves as a general model for a neutralizing agent in the concept of transformation of a diagnostic assay to a relative potency assay. The experiments described here in general, but I won't go through the details, as you will have access to the details a little later after the webinar, and I'll let you look at the details then. We'll move on and talk about the experimentation in more depth. I will call out a few specific parameters here, not so much in the equipment, but the MDCK cells. These are specifically chosen because there's a long history of those cells with the influenza vaccine, and it was actually, or with the influenza virus, it was actually patented and designed and optimized to produce this virus with the intent of creating vaccines. So it's been used here as the host for infection. The H1N1 virus itself is commercially available, but it is a custom reagent, and appropriate precautions and care should be used in requesting that and ordering it. So with that, I'm going to turn things over to Jessica and let her talk more about the technical details of the experimentation and the development of the methodology. Jessica?
Thank you, Jeff. So during the development of the relative potency assay, the WHO document, the neutralization assay, was used as a starting point. Utilizing that assay as a platform, the initial potency assay development needs were characterized in the cells as well as the virus. We looked at different principles and details in the WHO document, as well as creating sufficient robust assay, and looked at also qualifying that to regulatory standards. First, we performed experiments to determine the median tissue culture infectious dose for the TCID50 at various cell densities to verify the virus titer. Also, we took a look at the activity of the drug, the VIS-410, determining if a dose response could be attained, and if so, defined the dilution series needed to reflect the optimal reference curve. We focused on drug dilutions, the EC50, and the distribution of points in the curve. Other parameters examined during the development were timing of dosing of cells, assay plate type, preparation and incubation of the substrate, as well as other concepts we'll talk about shortly. Characterizing the cell culture is important to understand the long-term support of the assay. So we looked into split ratios, tracking confluency, viability, number of days in culture, how long they were in culture before being used in the assay, as well as monitoring the TCID50 over continuous culture. So starting with the WHO microneutralization assay recommendation of plating at 150,000 cells per mil, the TCID50 was found to be slightly lower than our targeted density virus titer. The TCID50 was then tested in parallel across various cell densities ranging from 75,000 cells per mil all the way up to 300,000 cells per mil, as well as across increasing cell culture passage numbers. With the higher seeding density, the experiments were spanned across 30 days in culture, and we were able to consistently get TCID50 results. So during characterization of the cells, it was determined that the cell culture performed best in the assay when passaged at least once from thaw and when passaged at greater than 1:10 split ratio. Now that the cell density and the virus titer were defined, where does the drug fit in? The drug inhibits the virus infectivity, therefore the nuclear protein of interest in the ELISA is not produced, and the absorbance readout should decrease as the drug concentration increases. The next step was to determine the drug's serial dilutions for the assay. In the next series of slides, we will step through some of the experiments conducted to determine the optimal drug dilution series. The graphs are all displayed describing the initial dilution of the drug from stock, the overall dilution of the drug in the assay, the design of the titrations, be it a 2-fold or 3-fold or custom dilution series, as well as tracking the EC50. Concentration of the drug is displayed on the x-axis, and the absorbance at 490 nanomolars is displayed on the y-axis of all the graphs. The first experiment looked in overall 1:8 dilution of the drug at the upper concentration of the curve. As the results show, at that concentration of the drug, the virus was inhibited and the sigmoidal curve was not obtained. You can see the lower concentrations of the signal does start to increase. The absorbance signal starts to increase. For the next experiment, we took those concentrations and used them as the starting concentrations, aiming to show a response without inhibition from the drug. Testing a larger serial dilution starts showing the dose response we are looking for, with an increased response in the drug at lower concentrations. However, reaching that upper asymptote was not achieved. Continuing to determine the range of the ELISA readout, whether we see complete inhibition versus the full response, we moved to a 9-point curve to optimize the optimal pre-dilution, final assay dilution, and serial dilutions across the curve to establish a robust curve with parameters that can be controlled, monitored, and compared. Some of the objectives and parameters trended were a stable EC50, stable upper and lower asymptotes, curve amplitude, as well as well-defined linear section of the curve. Keeping all these objectives in mind, experiments were designed to screen multiple pre-dilution ratios. Data demonstrated that pre-dilutions ranging anywhere from 1:4 to a 1:25 had reproducible responses. However, we were not reaching a stable upper asymptote to anchor the curve. When we started to look at 10 and 12-point curves with 3-fold titrations, as the upper asymptote of the curve became better defined, the EC50 began to shift as well to the left. Continuing the next set of experiments that were set up with 12-point curves with either lower pre-dilutions or 1:5 with varying titrations across the curve, we were able to find a sigmoidal curve that defined the dose response. So this leading the curves where sigmoidal were defined on the upper and lower asymptotes, as well as the curve with 8 points on the linear portion of the curve and 2 points anchoring both the upper and lower asymptotes for the final design. With curves that began to find the dose response, time was taken to begin to see the effects of other aspects of the assay and how they may impact the results. So the substrate for the assay was sensitive to light and heat. So due to the sensitivity of the substrate, preparation time before use, exposure to room temperature, exposure to light, and incubation time in the assay were all trended. Looking at different plates as well as a source for the assay, we looked at general tissue culture treated plates as well as specific coated plates recommended by the WHO assay. So we focused on how the raw data signal was impacted for all of those. There are various control signals between the plates. So if we look at the controls that were on the plates, we had cell control wells, which were just cells only and media. And so if we look at those difference between the tissue culture plate and the coated plate recommended by the WHO, the background signal is quite different between those. Sometimes you can see we had up to 2.5 times the signal in the TC plates. The EC50 was also affected and was observed to be 2 to 4 times higher, as well as the overall raw signals in the upper and lower asymptotes. So in summary, during development, we looked into parameters such as the cell culture. We looked at the density to optimize and obtain the TCID50 of interest, as well as characterizing the optimal culturing of the cells for use in the assay. We looked at substrate preparation and temperature exposure before use in the assay. We also looked into the incubation time of the assay, ranging from 4 and up to 20 minutes, finding that 4 to 9 minutes was optimal to keep the cell control wells below the recommendation of WHO reference of 0.2. We looked at different plates where the coated plates overall had a lower background interference in the raw data. And we also looked into the timing of plating the cells. So if we dosed the cells onto the plate before or after at the co-incubation with the drug and virus, we also saw similar results, however, increased variation with that order of operation. So having final conditions for the assay, the next phase was to test a range of nominal drug concentrations to ensure it meets the standards for routine implementation. So we moved into GMP qualification and validation to support the project. So during qualification and validation, the assay parameters that we looked at were linearity, accuracy, repeatability, intermediate precision, range, robustness, specificity, and stability. So just as a reminder of the assay design, the drug is performed in serial dilutions, is added to the assay plate, virus is then added, and they co-incubate for an hour while the cells are being prepared. So at the end of the co-incubation, the plate is dosed with 30,000 cells per well, and the assay is then allowed to incubate overnight up to 24 hours. The cells are then washed and fixed before proceeding with the sandwich ELISA and a colorimetric readout. The linearity of the assay was determined by examining the performance of samples at known nominal drug concentrations against the reference curve using both constrained and unconstrained curve analysis. So for the data that I'm going to present today, I'm going to show a % RP, or % relative potency data based on equivalency from unconstrained sample curves against unconstrained reference curve parameters. And I'm also going to present % relative potency, or % REP data presented from parallel line analysis, which is the comparison of the constrained curves fitted against each other. So the linearity assay was evaluated during qualification across 7 plates over 5 days and 3 different analysts, with a coefficient of determination of the regression line for the mean % RP of the NDC was 0.95. And during validation, this value was 0.98. As you can see in the graph displayed, the 125% NDC, which is demonstrated on the y-axis, is the target, actually the mean fell below the expected, and that was due to an outlier that we did not exclude from the data moving forward. We look at precision of the assay, and it was determined by looking into intermediate precision as well as repeatability. So just as a quick reminder, this graph does show both sets of data. So the % REP is based on unconstrained curved analysis, whereas the % RP is based on unconstrained curved analysis, and the % REP is based on the constrained curved analysis. So for repeatability shown from qualification, a minimum of 3 replicates at 3 different NDCs, ranging from 50 to 150%, by a single analyst show that the assay is capable of an overall CV well below the widely accepted value of 30%. The data also shows that the constrained curved analysis is a better determination of the target nominal drug concentration. The intermediate precision was assessed as the overall % CV at each NDC level, accounting for all factors such as plate reader, multiple plate washers, various incubators, analyst, cell culture passage, number of days of assays, etc. So for qualification, the % CV was around 17% and 15% during validation. So both of these are indicators of the utility of the assay to determine the relative potency within the range of the assay. Moving on to accuracy. The accuracy of the assay is the closeness of the test results determined by analysis to the known value of the sample. So once again, comparing the unconstrained and the constrained curved analysis, the accuracy of the assay is 98.9 to 125.6, and 88.5 to 109.5 respectively, with CVs of approximately 18%, well within the typical criteria at this phase of the assay of 70 to 130% and less than 30% CVs. Robustness tests the bounds of the critical assay steps and parameters. So, for example, cell density and critical incubation times. So for this robustness experiment, an increase of plus or minus 15% in the cell density and a plus or minus 15% in the critical incubation times were examined. So the lower cell density and the lower incubation times led to an overall lower raw data signals and relative potencies further from the known value. However, the results were within the variation seen within the assay. Short-term stability was also examined during qualification and validation. Stability was evaluated for variance of samples and virus handling. So we treated both the drug samples and the virus to room temperature, 2 to 8 degree holds, as well as freeze thaws. The assay was capable of identifying that when the virus was exposed to more than 24 hours at room temperature and more than 48 hours at 2 to 8 degrees C, the assay could not meet the control acceptance criteria. The assay was also tested to determine if it was capable of identifying degradation due to formulation or stressed holds at high temperatures and to explore the stability indicating aspects of the assay. Forced temperature degradation, exposure to high temperatures for 5 to 60 minutes, induced a clear shift in the dose response curve over time, suggesting that the assay could be stability indicating. Moving on to specificity, in this experiment, assay was performed in the presence and absence of the nucleoprotein antibody or detection antibody. The Gen 5 PLA plots demonstrate the lack of dose response curve, indicating that the sandwich ELISA will not work without the presence of both antibodies. And you can see that by the flat green line just slightly above the x-axis on both of those graphs. Additional specificity experiments recommended for further analysis could be to test a generic antibody to challenge the ELISA for specificity for the drug, as well as various formulation buffers' impact on the assay itself. At this time, I will turn it back over to Jeff for some conclusions.
Great. Thank you, Jessica. So to kind of wrap things up, what we've shared is that a microneutralization method here, the WHO method for H1N1, can be effectively transformed into a method for relative potency to determine that relative potency for a specific neutralizing agent, here the monoclonal antibody. We also described how the experiment was optimized to meet the needs of a GMP release assay and the parameters that were explored to get there, and then finally that the assay can be and was validated to current standards, including accuracy, dose linearity, range, specificity, robustness, and precision. Other attributes, including stability indicating ability, were also explored. In conclusion, the WHO protocol for diagnostic testing has been successfully transformed and optimized and is being applied to the VIS-410 neutralizing antibody. This represents a general and broadly applicable approach to in vitro potency analysis for viral neutralizing agents. The assay reflects the mechanism of action of VIS-410 by complexing with surface proteins. It neutralizes the virus and prevents infection. The assay uses quantification of the viral nucleoprotein, which is selective and provides a quantitative readout from the MDCK cells. The assay was optimized for critical attributes, including those listed, and the assay was qualified to current standards. This assay is very much aligned with the approach for the 3 Rs, reduce, refine, and replace for animal-based testing, and provides a very capable methodology and, again, generic transformational approach for antiviral agents and the determination of their relative potency. Finally, I want to acknowledge and thank again the team from Vistera for their support, materials, and scientific input. And I want to reemphasize the general nature of this approach. To successfully achieve this, translate a diagnostic microneutralization method to a relative potency assay, the requirements are to have an infectable cell, to have the neutralizing agent and have it well characterized, to have a source of virus that's reliable and to have a reference standard or reference material by which to compare. This is specifically applicable to the H1N1 here, but it is generally applicable to other viruses as well as other neutralizing agents. And then finally, I'd like to thank you for your attention, and hope you're all having a great day. Thank you very much.
Thank you for that very insightful presentation. I hope that our audience found it of great interest and value. If you do have any questions for both Jeff and Jessica, then please continue to submit them using the Q&A tool in the right-hand side of your screen. We have received a number of questions so far, so we will address some of these now live on air. So firstly, how do you determine the similarity between curves?
Thank you, Tristan. That's actually a really good question. So we looked at that in different formats. So we looked at both the constrained and unconstrained curve fits. So for the constrained, we looked at the F-test and determined if that met parallelism. And for unconstrained, we looked at equivalency ratios between the upper and lower asymptotes as well as slope ratios. We also looked at the difference in upper and lower asymptote ratios as well for the similarity between the curves.
Okay, and what are the critical attributes for this assay in order to achieve consistent relative potencies?
Thank you, Tristan. I'll take that one. The critical attributes are well-characterized cells and viruses. They must be matched to meet mode of action. And then, as was just alluded to in the question posed about the curves, having a robust system that produced robust curves, robust analytical data, based on that in order to provide the relative potency via comparison of those curves and the responses. It's also important for the neutralizing agent to have a well-characterized material and a consistent process to do so.
Okay, and how was the reference standard characterized?
Yes, so we took the reference standard and ran multiple experiments at 100% NDC as well to characterize the upper asymptote, the slope, and as well the lower asymptote, and kind of set ranges around what we typically see for that, and as well for the C parameter that's within that, and then we kind of constrained that before looking at other samples.
But there were other biophysical tests performed by our collaborators that we don't have the details of at this point.
And what are the key considerations to apply to other virus neutralizing agents, systems and modalities?
Very much related to the other critical attributes question. It's key to have a good cell line and to have one that can be robustly maintained and well characterized. Ideally one that's commercially available. And then also have a good source for the virus and the virus of interest, which is consistent, robust, and can be well characterized and maintained for the duration of the study and the development.
Okay, and you mentioned that one passage after FT was optimal. How much did it improve the assay?
Greatly. Actually, if you sawed the cells and then plated them at the first passage, the assay would not meet the control set for the cell control as well as the virus control for the assay. So the assay would not work unless you had at least one passage. After that passage, and it was continued to culture, and then they were harvested and plated, the assay would actually meet the control standards for the plate.
And did you evaluate different H1N1 strains during optimization of your dose response curves?
No, we only evaluated that one particular strain.
And what is the percent CV of the accuracy test?
The overall percent CV across the NDCs for the accuracy test was 18%.
Fantastic. And Did you arrive at acceptance criteria for the assay? And if so, what are they and how did you get there?
We did have recommendations for acceptance criteria based on the qualification and validation data. So there was acceptance criteria built around equivalency as well as evaluation for slope and upper and lower asymptotes and the cell control wells and the virus control wells. There was not an extensive historical data ran with this. It was, development, qualification, and validation. So over time, those criteria need to be reevaluated and trended and tracked. But there were some that were built around the slope ratios and the upper and lower asymptote.
And how did you characterize your virus stocks as a GMP critical reagent?
So from our side, the virus stocks were prepared by a third party hired by our client Vistera. So from our side, we would get aliquots of the virus, and then we would just confirm TCID50 as we would run the assay across the cell cultures based on comparing it to the C of A. All of the other criteria around that was maintained by either the maker, I believe was ATCC, or controlled by Vistera. In our hands, it was just tracking the storage temperature and the performance in the assay.
Okay, and what was the range of passages for this assay?
We actually showed that the assay could work anywhere from passage 1, and I believe we took it up to 30 days in culture under our development, and we never extended past that, and it was still working at that point. And I have to go back and look specifically for the passage number, but as long as it was in passage 1, at least through 30 days of culture post that.
Okay, and why was this cell line chosen?
This cell line was chosen for a couple of reasons. One is because it was part of the WHO assay, but the other is this cell line was specifically developed with H1N1 in mind and has patent protection as a manufacturing source of H1N1 vaccine. So had a great alignment with mode of action and a high probability of success in generating a viral response.
Okay, fantastic. Well, thank you very much, Jeff and Jessica, for answering our questions. Before we wrap up, do you have any last comments that you would like to add?
Again, just appreciation for the time and participation of those who asked questions and those who attended the webinar. This has been a great project, and we'd love to see this translated into other opportunities and propagated across other viral neutralizing agents. Thank you all very much for your attention and participation.
Fantastic. Well, I would like to once again thank our wonderful presenters, Jeffrey Patrick and Jessica Weaver, as well as you, our listeners, for your time and for your questions. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at bioanalyseszone. Don't forget to visit us at www.bioanalysis-zone.com for more webinars. Thank you again for attending, and goodbye.